Good afternoon, everyone. I hope we can begin. Good afternoon once again to everyone, whoever has gathered to join this meeting today by Professor Jose Remacle on the basics behind various DNA and mRNA strategies used for the development of COVID-19 vaccines. Dr. Jose Remacle is not a strange person to St. John's Research Institute. He's been an adjunct faculty for more than a decade, and most of us have been guided by him, and is also well known to many others in the Research Institute. But a brief introduction to Dr. Jose. Dr. Jose is a retired professor of biochemistry from University of Namur in Belgium. And during his tenure as a head of research unit in cellular biology at the university, he has guided several graduate, postgraduate, and PhD students, and also has had more than 200 publications, which have been cited more than 13,000 times. And most importantly, he's the inventor of 67 filed patents, 52 of which have been granted either in United States or European Union. He also has to his credit the notion of spinning off 3 companies through his academic and industry collaborations. Dr. Jose's main interest and area of research, however, is actually cellular aging, effect of free radicals on cells, and stress-induced accelerated aging. But what keeps him active to this day is his interest in technology development. And he has been the pioneer for the development of DNA and protein biochips in genomic and proteomic research. These have been extensively used for both diagnostic and prognostic use in both infectious diseases and oncology. Without any further delay, I invite Dr. Jose to start his talk. Welcome, Dr. Jose.
Thank you, Dr. Jyoti, for this two good introduction. So I will really start immediately. So I go there, yeah, and then share, and then forward. And then I will go to this one, okay? Is it okay? Okay. Can I perhaps remove on the top? So, in fact, I will try to give you science, but also to give a few ideas about general idea about the research and the relation between research and medicine. I started this talk. Okay, first, if you want the detailed information, I have made a website. The website is Vaccin, ARN and ADN, so this is French, but the first page, I have put everything in English. But otherwise, the next page are, oh, sorry, I have to remove my mask. So you have the English one on the first page. So if you want to go and read more into detail, there are much more, about 100 pages, you can have more detail. I cannot go into detail here, so I really want to stay on the concept. So the origin of this work was very strange. I had a discussion with my family doctor, and he said he spent the day answering the question about this new vaccine, RNA, DNA vaccine. All the patients, they wanted to know, they were scared and so on. So I decided to go because I also did not know anything about this. And I found that a beautiful research which was made for the last 20 years on the RNA and DNA vaccine that I did not know. And I will try to share a little bit with you, but I will try also especially to answer the question which was there on the newspaper, on the TV, and so on. Then I say it is our duty as a professor to have a social implication in public education. I just want to raise this point because I am here in the medical faculty, and I saw how the medical people came to TV, came to the newspaper, and really helped into the education of the public. I just want to raise this point in St. John's. I think you should have a reflection about, can you do something for public health education? What we have seen in Belgium, it was a retired professor who was coming all the time on the TV explaining with curve and graph and so on, what is happening, what is the variant, why do we have to close the school, why do you, it was really wonderful. And I remember one head of the ICU of the big university in Louvain, who came one day on the journal and said, look, we are 90% full in the ICU. If we continue like that, in 2 weeks, we have to refuse the people. And so we have to go for lockout. And I remember the impact of when you have the head of an ICU professor who come and say that on TV, the people believe. So I think there is a reflection to have as an academic, in medical academic, to about the duty you can do, not all the doctors, but a few of you, to be socially involved with the news. Okay, I also want to put emphasis, to really put first that as a scientific, I don't believe, I want the fact. And I just give you an example. This is a publication which was made in 3 universities, and they just looked at the amount of antibody in the patient. And if I just analyze this data, I can see here the people who have not been infected. If they have not been infected, you see they have no antibody against the COVID. If they get the first vaccination, after 7 days, you see a huge variability. They start to have IgG, but you see from one to the other, it is a huge variability. After 21 days, you see, then they start really to have all of them start to have antibody, okay? Now, if they get the second injection, after 1 week only, you see 10 times more than they are 7,000 units. So let's give you, for example, these are the data. Why do we have to wait minimum 21 days? You have the answer. You see, after the second injection, you have to wait 1 or 2 weeks. You are at the maximum. You see that if you have been, that's also something that you have seen. If you have the first vaccination, you can be reinfected. Sure, you can be reinfected, but you have already all the cellular mechanism has been put. So your lymphocytes are chosen, you have differentiated division of your lymphocytes, so you can react much faster. Now, if you look for the patient who have been infected, okay, without vaccination, this is the amount of antibody of infected people. And you see from the beginning, they are about the same amount of antibody as the first vaccination. It's about. And you see after 7 months, the antibody decrease. Even after 7 months, they still have quite antibody. Now, if you inject them with a vaccine, you see after 7 days, it shoots up, exactly like the second. So infection is exactly like the first reaction of a vaccine. It is the same. And if you give them one vaccine, they shoot up and they reach the same after 21 days, they see the rage level as 2 vaccines. If you give 2 vaccines, it doesn't change. So these are the facts. And then from there, you can explain what you see in the medical with your patient. Belgium is the world champion in vaccine, sure, we know it. Huh? They also play football very well, but they also produce vaccine. We have GSK, Thermo Fisher, We have Pfizer, Janssen Pharmaceutica, and so on and so on. So why do we have to give this information? Because, you know, there was a fear among the people. And fear in biology is a survival instinct. When you don't know something, you have the fear, and when you have the fear, you go in all directions. So it's becoming a non-rational thinking. So the only way to overcome the fear is for scientific leaders and doctors to explain what is happening. And then when you understand, you can go above your fear. So it was in the beginning, really, we need the doctor to come on TV to explain and so on, to convince the people they have to be vaccinated. So these are typically the questions I got from the doctor. What is RNA-DNA vaccine? How does it work? Why do we have to wait 21 days? Why it was so quick to develop? Generally, a vaccine takes 2 to 3 years. Here it takes 6 to 8 months. How come it can produce so much and so fast? Do this vaccine modify our genome? Can we trust what the pharma companies say? You know, this idea that the pharma, they just want to make money and so on. Can vaccine have serious side effects? And how long are we protected? These are typically questions which was well, normal. We have to try to answer. I cannot answer all of them in an absolute way, but at least give some information. Okay, so we will start with the messenger RNA vaccine. Okay, I suppose you still remember this slide. Information is in DNA, then we have RNA, then we have on protein. DNA to RNA is transcription, RNA to protein is translation. DNA, RNA information is nucleotide, AGCT for DNA, AGCU for RNA, and from RNA to protein, you change from nucleotide to amino acid. Why do I give this basic one? Because it is, you can understand vaccine. You can understand why do we have all these vaccines. If you are like Bharat in Hyderabad, you are producing vaccine with, in fact, it was not attenuated, inactivated virus, and you have the Covaxin. If you produce protein like hepatitis B vaccine, you will produce recombinant protein for the COVID. This is now Sanofi, GSK is at the end of their production. And now new came RNA. You can try to use RNA to produce the protein. And this is Pfizer, Moderna. You can try even to use DNA to produce RNA, to produce the protein, and this is COVID-Shield and Sputnik. So this basic biology show you that each company try to produce the vaccine with what they know. If they know protein recombinant with that they produce in yeast, they will produce. And only these two new vaccine, what was unbelievable for me, come from small research people laboratory, not from big company. And they went much faster than the big company. All the big company were beaten. Pfizer took BioNTech, and Moderna is Moderna, and the AstraZeneca took Oxford research. So it was basic research who led to the development of RNA DNA vaccine. Okay, no, it is not the correct one. Anyway, so and the transfer RNA tools, it is the RNA is the molecule which transfer the information from AGCT to amino acid. Okay, I go to the next slide, it will be more easy to understand. Transfer RNA is on one side the anti-codon. For example, you have tyrosine here. The codon for tyrosine is UAC. The transfer RNA is the complementary sequence AUG on one side. And on the other side, it is tyrosine. Okay, so this is the transfer of information from amino acid, from nucleotide to amino acid. So these little RNA, transfer RNA, are beautiful. So there are as many transfer RNA as there are amino acid, even more. So how does it work? So this is a machine called ribosome, and they can fix two messenger RNA. One messenger RNA, one transfer RNA, sorry, will come on one side and the other will come on the other side. You see here, there is already the amino acid making the protein. So how does it go? The tyrosine one will look at UAC, bind here. This is serine, and serine codon will be AGC. It will bind to AGC. And then when this is formed, ATP will come and transfer the serine into the tyrosine. So this amino acid will come from one to the other. Then this one will be free, it will go away. The ribosome will shift a little bit. So this one will become there, and this is free for the next amino acid. So this is the way the protein is sequenced, and it is done in a sequence which corresponds to the nucleotide information. Now, the protein can be released in the cytoplasm, but it can be released inside the endoplasmic reticulum and then go and export to the outside. How does it go? If you had what we call a signal sequence on the messenger RNA, you will have a small sequence protein there, which will be recognized by some protein of this membrane. They will bind, and the protein which is synthesized will go through the membrane, will happen into the endoplasmic reticulum, will be cut, and will be in the endoplasmic reticulum, where they can go to the plasma membrane and then export it outside. So you see, depending of how do you do the messenger RNA, you can synthesize inside the cells, or you can synthesize with export outside the cell. So the researcher will manipulate the RNA so that if they want to get it inside or outside. So this is an example of where the research can really help. What do you want to do with your vaccine? So now, can I change? I'm sorry, but this is an old presentation. This is not the correct one. I'm sorry, I will stop 2 minutes. Please, can you read this one? No, it's not this one. No, this one. Yeah, please. Yes. By the time it takes, do you understand it until now? Normally it is what you have studied in the school, no? So normally you should be able to understand. St. John's, yeah. Okay, now we got it. St. John's, open it. Yeah, St. John's short. Okay. Thank you. Okay, so we continue. Thank you very much. Okay, so now we are at what is the vaccine? What is messenger RNA vaccine? Messenger RNA vaccine is an RNA. So you have the RNA, you have the coding sequence. For this one, you have the protein of the virus, which is the spike protein S. So you put the S protein, and then you see you have regulator here called 5' UTR. Then you have regulator here called 3' UTR. You have a cap and poly. Okay, we go through this very quickly. So the central part is the sequence. It is to start with a start codon 3 and end with a stop codon. So it can be read by the ribosome. So this is the sequence of your protein. Now you have the 5' UTR, 3' UTR. They are regulatory elements which are very important because they will decide it, that the sequence can find the ribosome correctly. They can stabilize. They can be read more, longer, quickly. They can be more efficient and so on. So here we use for this, most of the vaccine messenger RNA, we use 5' UTR, which is untranslated region. UTR is untranslated region from the globin, human globin, because the human globin is synthesized for many days. That is why we will use this UTR from the globin gene. Then we have a cap. Why do we have a cap? If we don't have a cap here and a cap there, this is a chemical cap, this is a polyethyl cap, because the RNA can be quickly digested by RNAs. So we have to protect the 5' end and the 3' end of the RNA. So I went fast, but so all this has to be produced when you do the messenger RNA vaccine. You have to put all of this. How do we do? First, we have to find the sequence of the spike protein. If you are a little bit in the molecular biology, it is just playing on your computer. In one day, you find everything. You have two websites, one American NCBI and one European Ensemble. And these are the same website, but they have different software. And with a little bit of use, you can download and find the sequence that you are looking for. The software, I have made a website, qPCR site, where I explain how to use, for what you want to do, which software you have to do, and with the link to. It is easy. It's made for the researcher to find this type of, to find all this information. Why do I say that? Because that explains why they went so fast. You can do that in one day. You can find this sequence in a few hours. You can have everything. So it's very easy to do. Then, when you have the sequence, you have the sequence of the UTR from the globin. What you do is you restriction enzyme, and then you can attach both. This is also typically, I will explain a little bit later, this is typically a technique of the molecular biologists. They can, it's like copy-paste. They can do the same like copy-paste to put the sequence of the UTR from the globin on one side and on the other side. Once they have done this, they will put it into a plasmid. And a plasmid is a small, I think I have it here. Yeah. A plasmid is here, so it is a small DNA which is circular, where you can insert the sequence that you want to have. Like you cut with restriction enzyme, the restriction enzyme will cut here with this being open. So this is open, it's sticky end. So if you cut your plasmid with one restriction enzyme, for example, EcoR1, and you cut the sequence that you want with the same enzyme, you see that you can link it to each other and you can multiply. So it is easy to insert this, when you have this molecule, to insert into a plasmid. Once it is in a plasmid, you can multiply the plasmid, and then what do? You do now a technique that you know, which is called PCR. So the PCR will amplify the sequence that you want and will amplify billions of times. When you have amplified your sequence, you retrotranscribe into DNA, into RNA. So you have enzyme which copy DNA into RNA. The cap you just had as it's a chemical cap. You just make the reaction. And poly-A, you use a poly-A polymerase, and we put the poly-A. So you have your codon, you have the 2 UTR, and you have a cap, and you have a poly-A. You have your messenger RNA. So it's easy to do. In a few months, you can do that if you are a little bit of expertise. Just two, I mean, okay, that's a slide that you know, PCR, you have a DNA, you eat it, the 2 strands separate, you put small molecule called primer, they will copy. Again, you have 2 DNA sequence. Each cycle, and then you come back and do it again and again and again. So denaturation, we call it annealing, and then elongation, and you have multiplied by 2. What is the beauty of PCR when you do a COVID, but also when you do your messenger RNA, is that you multiply by 2 each cycle. What you have to keep in mind, please, is this. 10 cycle means 1,000 copy. 20 cycle, 1 million copy. 30 cycle, 1 billion copy. 30 cycle in 2 hours, you multiplied your sequence by 1 billion times. That explains why they can produce so much messenger RNA for the vaccine. So here I have explained why they went so fast, because it's easy. You just go on the website and you do your construction and you do your plasmid and you do your PCR. Why do you produce so much? Because of the PCR. So that explains why they went so fast, and you have so much of, I mean, so easy to make this vaccine, much more easy than the previous one. No, it's not the end of it. The messenger RNA is to go into the cell. There was many research on this topic. That's a beautiful topic. How to introduce this messenger RNA into the cell. The final solution which come out in Moderna and Pfizer is this lipid nanoparticle. What is lipid nanoparticle? It is really small, very small particle of lipids, and they use, and this is also one aspect that I will say, they will put cationic phospholipid, mean phospholipid which are positive. So the RNA will bind immediately by itself because it is negatively charged with the phosphate. So they can produce this emulsion. It's a kind of emulsion where the RNA is trapped inside this little particle. So what happened to this little particle? So you have your RNA, you have the lipo nanoparticle. It will be endocytosed by the cell. Then in the endocytose, you will have acidification of the endosome. The protease will be activated, will destroy the membrane. The RNA will be released. So the RNA will go through the lipid, go inside, will be released. Once it is released in the cytoplasm, ribosome will come, they will copy, and you have the protein. This is what you want. You don't have anything to do with the nucleus. It is not a genetic manipulation. It is not a gene manipulation. It is just you use the ribosome inside the cytoplasm, and you stay in the cytoplasm to produce the protein. So this answer another question, is it a GMO or whatever? No, it is not a genetic manipulation. It is just to use the ribosome, and you see the ribosome is the key factor in the cytoplasm, and there is no ribosome in the nucleus. It is in the cytoplasm, and they produce the protein. So this is the RNA. So it's not so difficult to understand. I think you should be able to understand if you have a basic biology, cell biology or biochemistry. Now we go to the validation. There was a lot of question about the validation. There is still question, I have still question in India for the validation. How do they do the validation? In Europe, we have European Medicine Agency, EMA, and in America you have FDA, Food and Drug Administration. They are very strict. They have their own, the way they want to test really all the procedure has been well done. So you have the typically phase 3, like any drug. Phase 1, do you have any toxic effect? Phase 2, in healthy people, does it work? Here they can test if you have antibody. If you develop antibody, that means the vaccine seems to work, and also no toxic effect. And then thousands of people, phase 3, on an entire population. The validation phase 3, you take volunteer, you inject the vaccine or you inject water, placebo. You monitor all the people for several months. You look which one has been infected. You verify by PCR test to be sure they've been infected. Then when it is finished, when the study is finished, you just classify the people between vaccinate and not vaccinate, and then you make a graph. This is the first which has been published, Pfizer, and look how beautiful it is. This is really the most beautiful picture. You have here the amount of days, 3 months. Then in blue, you have the vaccinated people. In red, you have the people who have not been vaccinated. You see in the beginning, 7 to 10 days, both of them are sick. You have sick people, and you see even if they're vaccinated, you have to wait. You see in the beginning, both are sick. Then after about 10 days, finish. It's finished. The one who has been vaccinated, almost nobody is vaccinated, is sick. This is 95% protection. So this is 5% here sick compared to this one. So you can say 95% efficiency. This must convince you that the vaccine works well. There is no discussion. When you see this type of thing in, I don't know how many, there was 40,000 people in this essay, you have to be convinced. These are data. This is not belief. I don't believe in the vaccine. No, but I believe in the data. Why do we do a double-blind test? Probably you have already known, because we need always a control. Why do we need a control? We need a control because in both groups, there was dead people, there was sick people. How do we know that these dead and sick people are from the vaccine or not from the vaccine? If they are dead, all the death has been checked by a medical doctor. There is a commission who look at the cause of the death. Usually 3 doctors are looking, is it linked to the vaccine or is it they had already some medical fight before. And then we compare always in the 2 groups. The conclusion, like I take is AstraZeneca, because this is your COVID shield. 2 people die in the essay in the vaccinated group. One was cardiac arrest and the other one was atherosclerosis, and 4 deaths in the placebo. They were not linked to the vaccination. They were linked by the fact that they were old or there were some problem and they died. So the number and type of problem that they found was the same in both groups. So why do I insist on this? Because people tell you, I heard that one person was very ill after being vaccinated. You can see that. And they say, oh, I don't want vaccination because I heard there was a death in the vaccination. That's not correct. That's why we need a control. So this is a mistake, and this is a mistake that many people, many journalists do all the time. You need a control. If you have 2 deaths in the vaccinated group, you have 4 deaths in the placebo, because in 6 months, if you have all people, there will always be 1 or 2 who will die, and you will always have the same problem. So please, as scientific, as medical doctor, we have to explain we need a placebo control. And when we need a placebo control, a lot of the problem is removed. I insist on that because we have a lot of discussion in our country and I think all over the world. It is not a genetic manipulation. I explain why. So Pfizer BioNTech was 95% and efficiency and was safe. Now, behind that, I will not go now into all the what has been done, but a lot of research has been behind for the last 10, 15 years. The cap, for example, the cap analog, there was a lot of cap. Does it stabilize better than the other one? There was a lot of work doing on the cap. The UTR, the regulatory element, there was a lot of work on the UTR. They've changed a little bit the sequence to have better stabilization. The poly-A, also they check different length of poly-A to increase translation or stabilize. They modify the nucleoside to have an increased translation. Then they have all the purification technique, because if you have double-strand RNA, you produce more cytokine, you produce more inflammation. On one side, it is good because you don't need adjuvant, but on the other side, you can have side effects. So you have really to, the purification technique, removing double-strand RNA was a big issue. And then you can optimize the codon and the sequence to increase the translation. All this you have is published and in beautiful research. I can just say beautiful research, and they succeeded, and wonderful. Just to tell you that before, why did they went so fast? Before they went on the virus COVID, they were already designing messenger RNA for HIV, rabies virus, HIV, Zika virus, influenza virus. So there was still already a lot of vaccine developed that we did not know, which were under either under assay or they were already developed. There was a lot of essay on medical vaccination against cancer, again leukemia-associated CMV, glioma, human CNV, brain tumor. So why they use messenger RNA into this cancer research? Because it's easy, so easy to produce, so easy to inject. You can inject in your tumor the messenger RNA. It will do the protein that you want. So if you want to have TNF, you don't need to inject TNF. You can inject the messenger RNA for the TNF. If you want to stimulate the immune system and for a protein which has been mutated, you inject the messenger RNA of the mutated protein. So there was a lot of assay already done in cancer with messenger RNA. I give this slide because we are here in a medical school, and my prediction, within a few years, you will see a lot of drug coming out with messenger RNA. I can tell you, I have my son and my daughter-in-law in big company, and they are already working on this. So it will come. The new medicine based on messenger RNA. That is why I spend perhaps too much time on messenger RNA, but I think this is coming in medicine. Okay, I will go a little, time is going on a little faster on DNA vaccine. DNA vaccine, you know it is AstraZeneca, is your COVID shield in India. Also, many companies are working, Janssen Pharmaceutica, the 2 Chinese, and then other one. Sputnik is there, the Russian, the Chinese, Indian, Belgium, whatever. So there is a lot of company working on DNA vaccine. What is DNA vaccine? What is the principle? So, okay, the principle is to use already a virus which is not harmful. This adenovirus, and this adenovirus, we will incorporate the gene of the spike protein, we will incorporate into the genome of the virus. And it's very easy. This virus has the first gene, early gene, is a transcriptional factor gene, E1A, E1B, and this gene code for a transcriptional factor which will activate all the other genes. So it doesn't need any promoter, and so it is immediately transcribed into protein. So if we remove this gene, the virus stops dividing, cannot divide the virus. They need this one to activate all the other ones. So what they did, very simple, they replaced this gene by the S protein of the virus. So you remove this one, cut, paste. Again, it is typically molecular biology. You cut here with restriction enzyme and you put your sequence. So you remove the gene and you replace by the S gene of the COVID. And so you have your vaccine. You have a virus where you have the gene of the S, which will be immediately transcribed and translated into protein. You don't need anything. And the virus cannot divide because they don't have the transcriptional factor. So you have everything. Only one problem. Only one. So this is the one. You have replaced the E1 gene by your gene. Okay? Now, what is the problem? The problem is that if you have removed E1 gene, the virus cannot divide. So how do you multiply your virus? How do you multiply your virus if they cannot multiply? I thought I had it here. Okay. So the way you do it, in fact, your virus, you multiply it into a cell, and in this cell, you have incorporated the E1 gene in this cell. So I will explain a little bit later. So the cell produces E1, and the E1 can then be used to multiply your virus. But I will come back later. I think I will come in the production. So I summarize. You remove your first gene and you replace by the S gene, okay? And you have your vaccine. Now, how does it work? You have your virus. The virus is taken by the cell. You have the endosome. The endosome break and release the virus, and the virus will bind to the nucleus and insert the DNA into the nucleus. So the DNA of the virus with the protein, the sequence of the S virus that you are interested will be in the nucleus. So what will happen? This DNA will then be transcribed into messenger RNA. Messenger RNA will go out of the nucleus and then be copied by the ribosome, and you make the protein. So you have the virus go into the cytoplasm, inject the DNA into the nucleus, and the nucleus, you do messenger RNA protein, and you have your protein. Then, yeah, so the messenger RNA is copied by the ribosome, and you have your protein, and the protein can be externalized, cut in two pieces, put at the membrane, and activate the lymphocyte. So you start your immunization. This is what you need. So, okay, I come back to this problem of how do you produce in large amount in the company. The answer is to grow the virus in a cell where you have put the E1 gene. The E1 gene that you have removed from the virus, you have it in the cell. You use this cell, X cell, and in this cell you have the E1 gene. So the E1 gene will allow the virus to multiply. So you can multiply this virus in these cells, but you cannot multiply it in normal cell because in normal cell you don't have this gene. So is it clear? So you have removed one gene of the virus, that's so that the virus cannot divide. To divide the virus in the production in the company, you use a special cell where you have incorporated this gene. And so this gene, the protein, which is a transcriptional factor, which allows the multiplication of the virus so you can produce the virus. This is a difficult process. Why? There was a lot of essay, really a lot of essay to get it work. And there was many problems. In the beginning, they use for malaria, HIV, influenza, hepatitis C, cancer, but not very effective. The level was 20, 30, 40%. So the researcher had to work a lot to try to understand why this DNA vaccine is not efficient. So one of the main problems, there was many problems, but one of the main problems is the choice of the adenovirus. Adenovirus is a human virus where most of the children, you are sick one day or another. It's not very serious. So we know this adenovirus, it is part of our sickness when we are small. What does it mean? That means we have antibodies against this adenovirus. So now if you use this adenovirus and you inject it in somebody, your antibody immediately will catch the adenovirus and destroy it. No vaccination. So that was the main issue. So the choice of the type, the serotype, what type of adenovirus do you choose is a key factor. So AstraZeneca, they made also a lot of work, and finally they choose adenovirus for a chimpanzee. So they choose, but my expectation is that there are in the human being heterogeneity of antibody, some people who still have a little bit of some of the antibody against this adenovirus. And if they don't have the first vaccination, they get it on the second vaccination. So that is the problem that they face. And for me, this is probably why AstraZeneca is not so good as the other one. Because they will use a serotype, human chimpanzee serotype. They think they are safe. I am not so sure. But anyway, they are not safe the second vaccination because they have already vaccinated. After 2 months, they revaccinate with the same virus, and you have the antibody against the adenovirus. So the second vaccination is probably not so efficient as the first one. That's my feeling. Perhaps I am wrong, but that's my explanation why AstraZeneca is not so efficient as, for example, you will see Sputnik. What is the, just Sputnik, take it Sputnik. What is the Russian form? They say, oh, that's a problem. We will use 2 different adenovirus. So they use the first adenovirus serotype for the first vaccination, and they use a second one for the second vaccination. That's very clever. So if you do antibody against the first one, you will have no antibody on the second one. And you will see the result of the Sputnik and AstraZeneca. So Sputnik is much better than AstraZeneca, and I think this is the reason. They struggle a lot to make it work. So it's a beautiful technique. Again, you need really basic research. There is no way around it. Then they have also optimized, they have removed one gene to put better, bigger gene and so on and so on. So, but these are all optimization of the genome. But the main issue for me, what I read was this adenovirus. Okay, so we have Sputnik. So Sputnik also published validation. So again, you have 1,000 of patients. You see again, 10 days, both are sick. After 10 days, you see if you have the Sputnik, you stop. If you don't have the Sputnik, you have the placebo, you go. And then you make between this date and this date, you have a little bit of sick people compared to this one. So this is 8% of this one. And so you have 92% efficiency. You see Sputnik, perfect. For me, it's perfect. When you see that 80 days, you see that Sputnik is very efficient. That is the validation is published. So what they use in America and in Europe to get their validation is it's public. They also, just to give you an example, that they really check very carefully. These are the amount of antibody in the people who have been vaccinated by Sputnik. The people who are 18, 30 years old, 30, 40, 40, 50, 50, 60, more than 60, you see they have all very good antibody compared to the placebo. But also, if you analyze the data, you see here, 2 people with nothing. So again, we are in medical field. Variability is there. There are people who answer very well, and there are a few people which do not answer very well. So you are human, variability is there. So you can always have a case or which doesn't fit the main one. But you see in general, all the vaccination in all age is good. So when they produce this result, you have to accept that it is good, and this Sputnik is good for all age, even more than 60 years old. This is a good validation. This is well done. Now, I'm sorry I come to AstraZeneca. This also published. This is what they have published to get their validation. And so this is 2 doses exactly the same. This is when the vaccine, and this is the placebo. And you see that efficiency was, I don't know, 60% or something like that. Now, they did something intelligent. They inject, the first injection was half of the dose. And they get a much better result. And I think it is linked to the fact that they produce less antibody in the first, so that the second one is more efficient. So when you put half the dose in the first vaccination, you have a better efficiency, and there you have more about 80% efficiency. So this is AstraZeneca, and as I say, my explanation is probably the fact that you produce antibody against the adenovirus. So AstraZeneca vaccine, no, there must be a problem here. 90%, 3 days old, 90% here. Yeah, okay. Now, with AstraZeneca, you have to see, do they assay for the people who have been, I come back. These are the people who have been sick, okay? When you take the people who have been sick, the efficiency is not so good. Now, when you take the people who are hospitalization, you see you get much better result. 90% with 2 different doses, 62%, 2 doses identical. Why do you get better result? That means that people who are vaccinated still get the virus, but they don't need to be hospitalized. So the vaccine is there. They can be infected, but they are prevented for hospitalization. So the virus, the vaccine is working, but not enough to prevent new infection, but work enough to prevent hospitalization. So according to which criteria do you use, you can get 60% or 90%. But it is good, hospitalization more than 90% efficiency, perfect. Side effects. I take AstraZeneca. I don't want to go in all this. So there was 3 cases of, I am not a doctor, so you just can read it, transverse myelitis, which was creating problem. It was taken by a medical team, and they say it's not related to the vaccine. You have 1 case of very high fever, And most of the disease were the same in vaccine and not vaccine. Again, it doesn't look like they have any side effect different in the placebo and in the, now we can discuss, there was some very rare effect which came up later on. AstraZeneca, just to give you, published on Scotland, 400,000 people vaccinated, and you see hospitalization 90%. So it is okay for hospitalization. They can still be infected, but they can be prevented for hospitalization. There were no data on the old people. So exactly what I said. Okay, time is going. I come back now, last one, Covaxin. Covaxin is a very classical vaccine. How do you do that? It's this inactivated virus. So you take a virus, you take a cell line, Vero E6, you cultivate the virus, you take out the virus, centrifuge, and then you treat by, usually, very often, there are different ways, but chemical agent. You treat by chemical agent, you kill the virus, then you made some adjuvant to increase, to stimulate the immune system, and then you put it into, you sell it. So this is very classical way to produce. It is 70 years old procedure to produce vaccine. So vaccine must work. You inoculate inactivated virus, chemically inactivated virus. Now, just a few words. In 1963, 42,000 children were infected by polio vaccine. Why? Because the vaccine was not 100% killing the virus. It was a huge problem. There was 56 children who died. So it was a huge problem. And so the WHO made rules really very strict. Why is it so difficult? You have billions of virus. You have to kill all the virus, not 99%, not 99%, 9999. You have to kill all the virus. So it is challenging. So it was a huge problem. It was a catastrophic effect of this virus. So it is a challenge. You have to be able to kill all the virus. You can understand that. Then you will say, oh, then I will put a huge amount of the chemical. So they use formaldehyde and propionolactone, so two molecules to kill the virus. Okay, good. So I will put a lot of formaldehyde. Okay, put a lot of formaldehyde, you will not get any antibody. Why? Because formaldehyde binds onto the amino acid. And so your protein will be completely changed. So you will not make good antibody against your protein. You will make antibody against formaldehyde protein. Propionolactone is a little bit better. It is binding of the nitrogen of guanosine, so it affects the DNA more than the protein, but it affects also a little bit the protein. So you cannot put too much chemical. So you have to kill. So that is the challenge of this Covaxin. You have to kill all the virus, but you don't change too much the virus. Otherwise, your antibody will not be good antibody. So just to tell you the challenge of the production, so you have an idea that it's not so easy. But it's a procedure which is there. There have been a lot of change, a lot of check, and so on. So normally you can trust it. So this Covaxin normally should work and should give you antibody. But I just want to make a little bit point on this production part. So it was Bharat Biochemical. So 2 days ago, it is a little bit shocking for me, 3 July 2021, they say they finally analyzed the phase 3 and on 130 cases. So I don't understand. Personally, I don't understand. They say it is efficient at 80%. But the preprint Covaxin is not available. I don't know. For me, it's a little bit strange that we have to believe them, and I don't want to believe anybody. I want the fact. So I will believe them when they give me the publication. So once the publication is out, we can really very clearly say, okay, it is real 78%. I hope they will release the publication fast. So Covaxin should work, 78%, 92% against severe symptomatic. So again, even if it is 80% of the patient, it protects almost completely against severe symptomatic. That means most probably everybody is touched by the vaccination. Some are not so well, but enough to prevent ICU hospitalization. So I think for me, if I have to summarize, I would say all the vaccine work, more or less. We have just to be, as a scientific, I want to see the data. That's okay. That's my mantra. I have to see the data to be convinced. Now, just a little bit side effect, just to give you an idea about what the researchers do, which is beautiful. So this is the virus. The virus binds to a receptor of the cell called ACE2, which is the angiotensin-converting enzyme, which is very strange. So the S spike of the protein binds to this receptor to go inside the cell. And you have bioinformaticians, and they did a beautiful work. So you see, this is the receptor of the cell. This is the virus, the protein of the virus. And they really could see where is the connection, where this S protein can connect with the AC2, with the receptor of the cell. And here you have seen, so the point here in red is the point of attach between the receptor and the S protein. Why do I give that? Because they can see in this sequence if some of this sequence can be different from the sequence of the protein we have in our body. That means if they are different, that means we can do antibody against this. So they found in the S protein, they found 34 places to make antibody, okay? From the 34 places, 7 to 10 are exactly there. So 7 of these places, we can make antibody against this place. We don't know exactly which one, but what we are sure, we will make many antibody because we have 7 to 10 of these, we call it epitope, where we can do antibody. Why do I give this result? I give this result to show you, you don't do one antibody. You do a spectra of antibody. Why is it so important? So you have probably many different antibody, many lymphocytes which has been activated. Why I say that? We will speak about the variant. Variants are coming. Variant is one mutation, okay? First, probably you mutate anywhere in any protein of the virus, okay. Now, suppose you also mutate the S virus. Now, you mutate anywhere in the virus. Suppose by any chance you mutate one of these amino acids here. Okay, one will be lost, but you have the other one. So the variant should be, the vaccination should protect you from the variant. From a theoretical point of view, I just speak about the theoretical point of view, you have enough different antibody so that one variant cannot stop the vaccination to protect. This is probably why all the vaccination protect against the variant. Again, it's a theoretical, it's a basic research, can try to explain what you see in the medical field. Okay, just for the dessert, new revolution in medicine. I already explained a little bit. Before that, a new research coming out in the pharma company. They don't only are interested in the messenger RNA, they are interested by the self-amplification messenger RNA. Self-amplification, what is self-amplification messenger RNA? Wow, they put another gene here. This is the gene that you want to do the protein. There is here another gene, which is, it is a replicase. It is they code for an enzyme called replicase, which can replicate the RNA. Means we can recopy the RNA. So with one RNA, you can make 10 or 50 RNA. So the RNA multiply itself. Wonderful. How did they found that? Okay, before, so this is exactly what here. So in fact, it is 4 genes. There is one gene, NSP4 with the replicase, then a gene for a cap, a gene for a poly-A. So with all these 4 genes, you can redo the full RNA. How does it work? So you have here your self-amplification RNA. Go into the cells. You have the replicase, which produce the replicase, the enzyme is translated into the enzyme replicase. What do the replicase? It will recopy the RNA. So you have multiple copy of the RNA, and here you have put a promoter so that some of the copy will be your messenger RNA. So it will copy full or it will copy one part. When it copy full, it will multiply the RNA. When it copy one part, it will copy your gene, which produce the protein. So you have two amplification. You have an amplification here, and you have an amplification here. Beautiful. So you have 2 amplification. So with very little RNA, because it's multiply itself, you will produce many RNA, and you will produce full RNA and little RNA. This is the new tendency for the researcher. I can tell you the first essay, phase 1, is a catastrophe because they produced too much. The amplification was too much and they had too much effect. So again, they will have to control, again, optimize. But okay, the idea was there. I dig a little bit, it's nice. Okay. Just to finish, personalized cancer research. So this is what is happening now. You start seeing the publication. RNA produce a protein. It's easy to do. In a few months, you can produce your RNA. 2-3 months, you can produce any RNA. You can easy to design, produce, low cost, very low cost. It takes longer. According to how you stabilize your RNA, you can produce protein 1 day, 2 days, 1 week, 2 weeks. It is locally inject where you need it. Now you need to find a situation where to introduce. That is the medical part. When you are in the medical, now the researcher, the technology can say, okay, I will produce for you. The doctor has to say which protein, how long, and so on. And then the scientific has to design the RNA for the doctor. It will come. So for example, I saw an article, there was a mutated protein on the surface of a tumor. They designed an RNA. Within 2 months, they re-inject this RNA to stimulate the lymphocyte, to stimulate the activation on the lymphocyte against the protein mutated means again the tumor. Very nice work which has been done. I don't know if it will work, but it is there. So if you have a tumor with a mutation, you sequence, you see a mutation in the protein, you can re-inject this mutated protein to stimulate the immune system. So this will come, I am sure it will come. You will not inject any more protein, you will inject the messenger RNA in the future. Just to give you, I copy paste BioNTech, the one who has developed. Look what he's doing. Therapeutic messenger RNA, express protein for therapeutic effect, individualized neoantigen specific immunotherapy, intratumoral immunotherapy, collaboration with Sanofi, here with Genentech. So you see, this one who has developed the messenger RNA, they are going full speed into cytokine, into whatever, collaboration with all the company. It will come. So I am sure in the future, in medicine, you will have to use messenger RNA. We need basic research applied to medical need, essential part of the medicine. Really, I think this is really explained that basic research is not a luxury, it is a need. For university industry, basic research, knowledge allow you to play in the game of the new product, be able to use them, be able to understand, see the parameters that affect the efficiency, adapt if necessary. You need to have the basic knowledge. Patent is not an issue. Patent is not an issue. I know the patent very well. I look at the patent of BioNTech and Moderna. There is no problem. You cannot protect messenger RNA. There was patent on some aspect, but you can go around it. For the moment, the field is open for developing a messenger RNA. Patent is not an issue. Knowledge is the issue. Just to show you, I like this one. So they are kissing the virus, we call it that kiss. Two lymphocytes kissing the virus. Beautiful, no? Okay. Good luck and good work. I hope you understood a little bit.
Thank you very much, Dr. Jose, for that elaborate presentation. I hope everyone has been benefited to a huge extent by this. We can now keep the house open for questions. The first option will be for the listeners who have joined in through the Zoom to ask questions. Either you can type your questions in the chat box or you can ask the question by unmuting yourself.
Hi, Jyoti. This is Sridhar. Can I ask Jose a question?
Yes,
sure. Sridhar, please go ahead.
Nice to meet you.
Yeah, hi, Jose. Enjoyed your talk. I had one or two comments about things you said and then a question. The comment was about the Covaxin trial. I happen to know some of the backstory of the Covaxin trial. The Covaxin phase 3 trial was published only on the 3rd of July because actually the Covaxin trial got started, the phase 3 got started towards the end of 2020, early 2021, when, as you know, what happened in India, the cases just disappeared. And this was actually an event, they enrolled 25,000 people. And this was an event, so the number you put up was actually the number of events they needed to see before they could close the trial. So those many events never happened. So they never could do the trial. So they waited for April, May, and only then they could complete the trial.
Okay, very good.
So that is the short answer. I mean, we can talk about the details. That's not, I think, very germane, but that's the explanation. But I had you spoke of 3 kinds of vaccines, all very interesting. But I was actually intrigued very much by this company, Novavax, which is now working with Serum Institute to make its vaccine. And their strategy is just to make the recombinant protein and make a nanoparticle of it. without knowing anything, because, you know, mRNAs were never authorized for vaccine before this. I thought that making a recombinant protein would be the simplest way of doing it. And do you have any comments? Do you think that it is not a good idea, or what are your thoughts on just the recombinant protein?
This is the best one, I agree. And I hope it will come as fast as possible. We have a disaster in Belgium because we have GSK with Sanofi, and they started the recombinant protein because it is the more safe. And there is absolutely no question about this. It is very well known. They know how to do it, and all the purification and so on. And the disaster was that the Sanofi made a mistake into the level of protein. So the efficiency was only 55%. They did not put enough protein. Oh. And they had to redo completely the phase. Okay. So it will come also, yeah, sure. If you can do this recombinant protein, absolutely, no discussion. This is the more safe and the best one for me. Now that I have dig a lot about messenger RNA, I would not remove anymore the messenger RNA. But you are absolutely right. The big company like Sanofi, Merck, and so on, they went for the recombinant protein. Absolutely.
Thank you.
I invite Dr. Anil Vasudevan, who has been monitoring the chat box, to ask further questions to Dr. Jose.
There's one question. Okay, sorry.
Come see.
So one of the questions is to know whether is there any ideal time for vaccination in a person who has just recovered from COVID? Can they?
You mean after COVID infection?
COVID vaccination.
What I can tell you is you have seen the antibody, and anytime. I think for me, the people, what I have seen, but okay, from a basic point of view, what I have seen is the level antibody in the people who have been sick is the same, almost same range as the one who have been vaccinated once. So I would do the same range as the vaccination.
Yes.
I don't see, but no, again, it is just based on the antibody level. I don't see why they should be 2 times, but at least once would be very useful.
Yeah.
You see the antibody
yes.
Speed up 10 times, 10 times more. The second vaccination or the vaccination after being sick, you increase 10 times your antibody. That means your IgG is there. When the virus comes, it will be bound by the IgG. So I would, from a basic point of view, now you are a medical aspect, but from a basic point of view, I have the feeling that vaccinate at least once the people who have been makes sense.
Right. I mean, so in summary, I think all those who have recovered from COVID also needs to take the vaccine. And by the government policy in India, now they have moved from 2 weeks to 12 weeks interval after recovery, and that's for various reasons they have made this.
But that is mostly because they want to save the vaccine.
Yeah, and also.
I don't see any problem.
Right.
Only the fact that if you don't have enough antibody, you can be reinfected.
Yeah.
That is the only problem. I suppose you see it now in India, reinfected people. That's what I understood. You have quite some people who have been reinfected.
Right.
And this is probably because they don't have enough antibody.
Yeah.
You see after the vaccination in 1 or 2 weeks,
the antibody response
very low, huh?
Yeah.
Very low. So I expect it, but it's just an expectation, the way it goes.
So,
is it okay?
Yes.
No, you have to save the vaccine everywhere in the world. In Europe, we did the same. AstraZeneca, we wait three months because they want to vaccinate as many people as possible. It's not a medical decision. I would say it's
that's why I mentioned
it makes sense. But if you, for me, after 3 weeks, 1 month, it's okay.
Thank you, Dr. Joshi. This is Neha Vyas. I have 2 questions for you. One is that given that adenovirus infections are very common in the early age of humans, what are the chances that the E3 transcription factor would be integrated in someone's genome? And when you give the adenovirus, the patient, I mean, that vaccinated person is going to make a lot of virus on his own, because that's the only missing point.
You are correct. What can I say? It can happen. I hope so. I hope it will not happen, but theoretically it can happen.
Okay.
Yeah, you are right. If you have any example, I don't have any example that it happened.
So is the severe sick case that you discussed about one such example, and hence now the adenovirus was somehow making them sick? I don't know.
Yes. Adenovirus, I have problem with adenovirus because I have seen so many publications. They've tried so many things. So it works, but it has to be optimized. And as I always say, human beings, you know, the heterogeneity is there. And I suppose some people have more antibody than other ones, some are more resistant. So anything can happen. I would personally prefer, like Dr. Sridhar said, recombinant protein or messenger RNA or attenuated or inactivated virus than the DNA. It is working, but it has to be really optimized. And if you read the old article, they have tried so many things. It was 40%, 50% efficiency. They had really a lot of problem to reach 80, 90%. Okay, they got it. I would do like the Russian. I would use 2 different adenovirus for the first one and the second one. When I see the result of the Russian Sputnik, I say, okay, it looks like it works.
So that is the other part of my question, is that the first vaccine, if it is a DNA or co-vaccine, nowadays a lot of people are talking about mixing the vaccines. So if the Covishield, after Covishield or co-vaccine, if you take Sputnik, do you think there could be issues?
That would be better. Yeah. It would be better because the antibody on the one will not react on the second one. Theoretically, again, theoretically, it should be better.
May I add a comment here? Actually, last week, Germany collated all of the results of people who had taken Covishield as their first dose, the DNA vaccine, followed by Pfizer. And they showed that actually the titers were closer to 2 doses of Pfizer than 2 doses of Covishield.
Perfect.
So there's data now, there's data.
Okay.
Perfect. It fit with the expectation. Perfect.
Yeah. So mixing is not an issue.
Yeah, no, it's fine.
No, to repeat with the same adenovirus is a problem. Yeah. So if they can avoid that by mixing, I would say, again, from a theoretical point of view, Dr. Sridhar data is wonderful because it substantiates the fact that you can shift from one to the other.
And do we stop at 2 doses, or do we continue like flu viruses every year, one vaccine? I don't know.
God knows.
Okay.
No, it's a good question. You have seen 7 months, you have a decrease of the IgG, but your memory cells are still there. So your lymphocyte and memory cell is still there. Do you need to reactivate them after 1 year? I don't know. I don't know. My feeling is that you will not need 2. Perhaps you need once to reactivate, Perhaps you will have to put a little variant, perhaps 2 messenger RNA with two or three variant, 3 or 3 messenger RNA, which can cover the variant. I don't know what they will come with. I would be on a messenger RNA. I would put in 1 year not one messenger RNA, but I would put a combination of messenger RNA with the mutated virus, so that you can. That's what I would do. You can broaden up a little bit and get a better protection against the variant. That's what I would do, because it's not difficult to do. You just have to change the sequence. You know, it's not easy. It's not difficult to do. Yeah, good question, but I want data. I'm sorry, but I want data. I had enough discussion with some people about vaccination. I just say stick to the data. That's my philosophy now.
So there's one question in the chat box. Do you have any data if these vaccines confer immunity to the fetus if the pregnant women are vaccinated?
Oh.
And does it differ for the different types of vaccines?
IgG pass in theory, the IgG goes into
yes,
the...
I mean, so the placental transfer is of the immunoglobulin G. I mean, it's most transferred most efficiently, and that's one of the way of mechanism. Yes, the G.
So it should
and of
go.
These, IgG1 is the most efficiently transferred. So that's one of the mechanism where the neonates are protected against many of the.
I think I saw an article about pregnant women, they can be vaccinated.
Yes, yes. A lot of countries like Australia, Canada, and New Zealand have been vaccinated. And India also has released guidelines to vaccinate.
It will not affect the fetus. Yes. And I suppose, I don't know, for influenza, for example, do the antibody goes into the baby?
Most of the maternal transfer of immunoglobulin G happens.
So it should be
passive
the same.
Immunity that happens.
So it should be the same here. The baby should be protected. And I see also I was thinking about coming here that, I don't know, in Bombay also, they show that the children, 50% of the children had antibodies. Yeah, so I say perhaps the children will live with this virus. Will have less, when they grow out, they will already have the immune system, which is used like we have for influenza, or I don't know what will happen with this virus. Will it disappear from herd, or it will be mutated all the time? I don't know.
There are too many unanswered questions.
Question, huh?
Yeah. So there's another question that SARS-CoV-2 virus is very similar to the SARS 2002 virus and the MERS virus, Middle East Respiratory Syndrome virus. So what are the major changes in the virus that vaccines for SARS 2002 and MERS cannot be effectively used against SARS-CoV-2? If I understand, the question is the vaccine against SARS 2002 and MERS cannot be used effectively against SARS-CoV-2. So they just want to know what's the difference in the
please. Oh yeah, because I don't know. Yeah, no, go ahead.
So what I have come across as an information is that the SARS-CoV and SARS-CoV-2 have mutations. Between them, there are mutations in the antigenic epitope. And so the receptor binding abilities, those mutations basically don't allow the antibodies which are raised for SARS-CoV-2 to act against SARS-CoV to act against SARS-CoV-2. So the antigenic epitopes are different, and hence the antibodies don't react.
They don't react.
They don't react.
Are there any more questions for Professor Jose? So there's one more question. So this question is, can there be another yet unknown sequences in the body which would be similar to the sequence we see when a virus binds to AC receptors at the interface? And if we start to create antibodies against it, won't it cause side effects which will be significant in the long run and may not be traced back to the vaccine? So We have a lot of undergraduate students who have joined this talk, and this is a question from one of them.
So the question is if we have a sequence.
So the question is if the sequence is similar to the sequence we see when a virus binds to the AC receptors at the interface. So I mean, if I understand the question correctly, it's like binding to something similar, homology protein receptor, the spike protein, binding to and then creating an autoimmunity kind of or an adverse reaction.
I would say probably not. If you have seen just the essay on with the biotechnology, the way the model of the S protein binding to AC1, they found so many differences. So they compare to all the human genome protein that we have, and they found, I told you, for the S protein, 34 different epitope which are different. So I don't think there is something in the body which is similar. I don't think so. Okay, no, anything can always happen, but from what we know, it doesn't seem like. Now, perhaps I just can add, for example, the AC2 receptor is transforming the that I did not talk about this. It's transforming angiotensin 2 into angiotensin 1,7. So the AC2 receptor destroys angiotensin 2. Angiotensin 2 is a vasoconstrictor. So if you block the AC2, you can have vasoconstriction. That can explain sometimes constriction of the tissue. And then you can also, if you block AC2, activate some part of the complement. And that there was a lot of study on that, because that explains why in the lungs you can have clotting of the blood and you can have vasoconstriction. Just by blocking the virus, when the virus blocks the AC2, ACE2, you can have these two effects in the lungs. That is how the doctor explained what they have found in the clinic about the clotting and vasoconstriction in the lung. Again, it is a correlation, but it looks like it is what is happening. So the fact that it is AC2 angiotensin converting enzyme explains some of the effect of the medical observation.
So now the questions are coming in. So maybe we'll take one last question. So your final verdict, which is the most efficacious vaccine? Is it Covishield, Covaxin, or Sputnik? I'm not finding the Pfizer or Moderna in here. It's among the three.
No, I'm sorry that you have seen that all the vaccine work. And they all work until, if you take hospitalization, they all reach 90%. So I don't want to say anything about that. What I would like to see, I would like to see medical students to be involved into research. That I would like to see. I started my PhD in a medical lab where all the researchers were medical doctors. And they really wanted to understand biology, what it was. And I found we need this medical doctor also into basic research to really understand what is happening into medical field. So I call as an old professor, I hope some of you will be interested to go for basic research in medicine, understanding the problem, trying to improve. This is what we really need in India. We need that. Not in America. Don't go in America. Stay in India and work here.
So, yeah, thank you, Professor Jose, for very much for your excellent talk and providing us a basic review of the various types of vaccines.
It was basic.
And I think for the undergraduates, what Professor Jose did was to combine the basics of biochemistry and other molecular sciences And I think it's a good reinforcement that the subject that you study in the first year, this is how we apply in a clinical setting and also for a clinical effect. So what we call as applied biochemistry, applied physiology, and applied pharmacology. And it's important to have a very good basic understanding of these subjects. So, and one more point for all those who have logged in, Professor Jose has a website, and there is a lot of content and information on the vaccine, and we'll be sharing it, circulating it in the group mail. Whenever you're free, you could access and go through the website. And if you have any further queries, we will also drop in the mail ID in which you can ask further questions. Thank you. Thank you all for logging in. And I'd like to thank
Dr. Jyoti,
the Molecular Medicine for hosting, Dr. Tony Raj, the Dean of St. John's Research Institute, for allowing us to disseminate to the entire campus. Have a good day. Thank you.
Yeah, thank you. Thank you, Dr. Jose.
Pleasure.
Very nice talk. I learned so much.
I don't know.
Thank you.
I try to mix.
No, nice. Good awareness.
Nice. I was fascinated. As an old professor, I decided not to work anymore, but one